Cell biology is a fascinating field that focuses on the study of cells and their intricate processes. In the realm of medical sciences, a solid understanding of cell biology is crucial for comprehending various diseases and developing effective treatments. In this script, we will delve into the complex world of neuronal cell communication and synaptic transmission. We will explore the mechanisms behind ion channels, membrane potential, action potential propagation, neurotransmitter release, receptor effects, synaptic integration, neurotransmitter recycling, neuromodulation, synaptic plasticity, neurotransmitter disorders, pharmacological interventions, and advanced molecular mechanisms. By the end of this script, you will have a comprehensive understanding of how neurons communicate and transmit signals, paving the way for further research and clinical applications. Neuronal cell communication is a fundamental process that allows neurons to transmit information throughout the nervous system. At the core of this communication is synaptic transmission, where electrical signals are converted into chemical signals and vice versa. Neurons communicate through specialized structures called synapses, where the axon terminal of one neuron interacts with the dendrites or cell body of another neuron. Ion channels play a crucial role in neuronal cell communication by regulating the flow of ions across the cell membrane. The movement of ions creates changes in the membrane potential, which is the electrical charge difference across the cell membrane. These changes in membrane potential are essential for generating and propagating electrical signals in neurons. Action potentials are the electrical signals generated by neurons to transmit information over long distances. The propagation of action potentials relies on the opening and closing of voltage-gated ion channels along the axon. This allows the action potential to travel from the cell body to the axon terminals efficiently. When an action potential reaches the axon terminal, it triggers the release of neurotransmitters into the synapse. This release is facilitated by a series of intricate mechanisms involving calcium ions, synaptic vesicles, and fusion with the presynaptic membrane. The precise timing and coordination of these processes ensure accurate transmission of signals between neurons. Neurotransmitters released into the synapse bind to specific receptors on the postsynaptic cell, initiating various cellular responses. These receptors can be categorized into ionotropic receptors, which directly control ion channels, and metabotropic receptors, which activate intracellular signaling pathways. The binding of neurotransmitters to their receptors plays a crucial role in determining the excitatory or inhibitory nature of the postsynaptic response. When neurotransmitters bind to postsynaptic receptors, they generate postsynaptic potentials, which are changes in the membrane potential of the postsynaptic cell. These potentials can be either excitatory, depolarizing, or inhibitory, hyperpolarizing. The summation of these potentials, along with other factors like spatial and temporal summation, determines whether an action potential will be initiated in the postsynaptic neuron. After neurotransmitters have fulfilled their role in synaptic transmission, they need to be cleared from the synapse to prevent continuous signaling. Recycling and reuptake mechanisms ensure the efficient removal of neurotransmitters from the synapse, allowing for precise control and modulation of neuronal communication. In addition to the direct effects of neurotransmitters on postsynaptic cells, neuromodulators also play a significant role in shaping neuronal communication. Neuromodulators, such as dopamine, serotonin, and norepinephrine, modify the excitability and responsiveness of neurons, exerting longer-lasting effects on synaptic transmission. Synaptic plasticity refers to the ability of synapses to change their strength and connectivity in response to activity. Long-term potentiation, LTP, is a form of synaptic plasticity that involves the strengthening of synaptic connections, resulting in enhanced signal transmission. LTP is believed to be a cellular mechanism underlying learning and memory. Imbalances in neurotransmitter levels or dysfunction in neurotransmitter systems can lead to various neurological and psychiatric disorders. Disorders such as depression, schizophrenia, and Parkinson's disease are characterized by dysregulation of specific neurotransmitter systems. Understanding these disorders at the cellular level aids in the development of targeted therapeutic interventions. Pharmacological approaches are widely utilized to modulate neuronal communication and treat various neurological disorders. Drugs targeting specific neurotransmitter receptors or affecting synaptic transmission can help restore normal function in affected individuals. These interventions often involve the use of agonists, antagonists, or drugs that modulate neurotransmitter release or reuptake. Advancements in molecular biology techniques have allowed researchers to delve deeper into the intricate molecular mechanisms underlying synaptic transmission. Current research focuses on understanding neurotransmitter release machinery, the role of intracellular signaling pathways, and the impact of genetic and epigenetic factors on synaptic function. In conclusion, neuronal cell communication and synaptic transmission are complex processes that rely on the precise orchestration of various cellular and molecular mechanisms. Through ion channels, neurotransmitter release, receptor interactions, and synaptic plasticity, neurons can efficiently transmit information within the nervous system. The understanding of these processes has significant implications for the diagnosis and treatment of neurological disorders. Further research into the molecular mechanisms and advanced topics in synaptic transmission holds promise for more targeted and effective interventions in the future.